Yeah, so as has been said, I'm a professor of biological systematics at Lund University and also the director of the Biological Museum here. And I use a lot of genetic resources in my work. I'm interested in the phylogeny and evolutionary history of Lepidoptera, so I sequence things from all over the world. And Nagoya, of course, has a huge impact on my research. So I've been tasked by the rector of Lund University to be available to other researchers to share my experiences, and talk about Nagoya issues with people who are bumping into these things. And by the way, should I turn on my camera? Or am I just a disembodied voice coming out from...
No, you're welcome to turn on your camera. We can see you in the corner.
Right.
So those people see you.
Turn on start video. Great.
Yes.
Yeah. Here I am. So what I thought about telling you is the steps that one has to go through. Now, I guess Johan and Sebastian didn't tell you what Nagoya is. So Nagoya is all about the access and benefit sharing of genetic resources from countries other than the country where you are doing your studies. So for us, if you want to sequence a butterfly or a protist or a plant or whatever from another country than Sweden, then you have to look into whether Nagoya Protocol is relevant for you. So there are 5 steps that you have to go through. You have to investigate applicability, then obtain relevant documentation, sign agreements, register and preserve all documentation with your university registrar, and report due diligence in the EU Digital to Declare, especially if you have EU funding. So I'm going to go through these steps one by one and give some of my experiences or what I've heard from others regarding each of these steps. So the first step is to investigate applicability. If you're going to go to Denmark to sample a couple of flies, does Nagoya pertain to you? Temporal scope. So Nagoya came into force on October 12th, 2014. So everything that has been collected after that date is under Nagoya. Everything collected before that date is not. Material scope, what is it you're going to be studying? What is a genetic resource? This is a big debate. Politicians have their own view. We scientists have our own view. But in general, genetic resources have everything to do with DNA, of course, RNA, of course, proteins, anything that's coded by DNA. So if you're interested in protein functions and so on, they're under Nagoya. But where does it stop? Some countries say that all the way to the phenotype is under Nagoya. Malaysia, for instance, I heard when I was visiting there that if you want to go into a collection in Malaysia and take pictures, you need Nagoya permits for that. That's taking a bit too far, but hey, each country has their own laws. Another thing you want to look into is whether you're going to be using existing collection. Are you going to go into a museum and access genetic resources from specimens in a collection? And if that's so, does Nagoya pertain to you? So this is a very, very difficult question because you might be interested in a sample that was collected in Democratic Republic of Congo 100 years ago, and you are going to extract DNA from it. When was that genetic resource accessed? Was it accessed when it was collected 100 years ago? Or was it accessed when you took it into the lab and extracted DNA now? If the interpretation is now, when you extracted the DNA, then you need Nagoya issues. If the interpretation is that it was accessed when it was collected, because it was collected to be studied, then Nagoya issues don't pertain. Keep in mind, all of this there's no precedence here in Sweden. Nobody has gone to court yet. So we're all sort of experimenting and testing and seeing when we'll be the first person who will be fined or put in jail for using specimens without taking Nagoya into account. Here is the most recent map of the countries which are parties to the Nagoya Protocol. So you'll notice that there are countries that are not party to the Nagoya Protocol. That doesn't mean that you're free to go there and collect. These countries will have their own laws. For instance, Colombia has very strict laws on collecting genetic resources in their country, but they're not part of Nagoya Protocol. What this means is that, okay, for EU-funded projects, yes, you have to deal with your permits and so on from Colombia, but you don't need to show the EU that you have followed the Nagoya Protocol. But you better follow the laws of Colombia, because jails in Colombia are not very comfortable. So you can see the vast majority of the world are part of Nagoya. And down here, you can see the website where you can find information for each country. So this is the ABS Clearinghouse website. And on this website, you can click on each country and you can find out about the laws for each country. Now, many countries have signed the Nagoya Protocol, are party to it, but they don't have laws yet. One good example is China. I have a colleague here who recently got herbarium samples from Harvard University. They were collected in China some years ago. The Harvard person said that they had all the right permits, so they could collect and take the specimens out. But now my colleague wants to sequence these specimens and asked me, so does he need to have the permits, or did the Harvard people, did they already get the permits? In this case, when we looked at the webpage for China, it says that China does not have laws yet in place for accessing genetic resources. So how do you interpret this? You should ask a lawyer, not me, but my interpretation would be that if the permits were in order for taking the specimens out of the country, then they're legal specimens and you can do what you want with them. There are no laws for accessing genetic resources in China as yet, but they might come tomorrow, next year, in 10 years, who knows? So you have to keep your eye on this website every time you think about accessing genetic resources from another country. Once you have decided where you're going to go, then you need to get the permits. So there are two kinds of permits. You need to have a so-called PIC permit, prior informed consent. You need to be able to, you need to tell the country where you're going to what you're going to do with these samples and why you want them and what samples you want. And they will give you a permit. This is like the old collection permits that many countries have needed for already a long time. And so you have your PIC, your permit to collect, your permit to export, and I think the next presenter will talk about the details of how to get export and import permits, and so on. And what you need to know is that your PIC is only for you. You can't give your samples to somebody else. That person would then have to get their own PIC, even if it's the very same sample that you collected for that very purpose. It's in your name, unless you have put in a clause that other people can work on these samples as well, and if the country of origin agrees to that. Then you need another piece of paper with a country of origin, and this is the so-called MAT, mutually agreed terms. What are you going to do with your genetic resources? Are you going to develop a product in pharmaceutics? Are you trying to make a product which will eventually be giving profits? In that case, you need a very clear contract with the country. What happens with the profits? You will have to share the profits in the way that you agree with the country. Many of us are doing basic research, so we're not looking to make profits. But the country of origin might say, okay, you're going to do basic research. You're using our genetic resources. You have to be able to give us something back. It could be by preferably by collaborating with a researcher in the country of origin. Resources helping, giving a course in your subject in the country of origin, inviting postdocs or PhD students or whatever, some kind of agreement with the country of origin. Now here I can tell you that in Europe, most countries will say, nah, we don't need a mat. You just, as long as you're doing fundamental basic science, you just go ahead. But you need that email to show your funders that you've done your due diligence, that you have found out that, okay, I don't need mutually agreed terms. I might need permits to collect. If you go to France, you'll need some permits to collect. And France will say that, oh, if you're just interested in taxonomy and systematics, you don't need a mat. But if you're interested in how the gene functions, then you need a mat with France. Okay? So that's the way it is. You need to figure out. Every country is unique. Keep that also in mind. Every country has its own laws. There's no one law. There's no one Nagoya Protocol. Every country has interpreted it in their own way. So you have to figure out for every single country where you want to go, how they deal with Nagoya. Very important. Keep track of all the paperwork. Keep and register it in the university registrar, what in Swedish is called diarieföra, the papers, and preserve them forever. Anybody can check on it at any point. And so this is very important. Make sure the paperwork is preserved. And finally, if you are on EU funding, then you need to have everything on Declare. This is a portal by EU where you can just keep track of everything, all your pics and mats and all your emails that you've sent and have never gotten a reply to. Those can be placed in Declare to show when EU doesn't audit that you've done your due diligence, that you have figured out everything that you need to do to be able to work with your samples, to access the genetic resources of your samples. So it sounds quite daunting, and in some cases it is daunting. There's no question about that. If you want to go to Africa, if you want to go to South America, if you want to go to Asia, prepare yourself for a lot of bureaucratic work. Within Europe, it's easier, fortunately. And what I can say for my own work is that I am turning more and more to Europe for my research, even though my research is basically global in scope. But the idea that I would have to go to 20 African countries in person to get all the proper permits to sequence samples that I already have, I don't have time or funding to do that, and the chances are that I wouldn't get the permits for everything, and it's just a nightmare. So for me, I'm just giving up on the rest of the world, and I'm mainly concentrating on Europe, where things are can be handled. Even though there are countries in Europe which are also difficult. Try to go to Greece and try to find the right person there. They have signed Nagoya Protocol, they have laws in place, but you can't find the right person to help you with getting the permits. I have a colleague here who has had this problem. And so, yeah, so that's what I wanted to say in this short time. I'm sorry I'm not there. It would have been interesting to discuss your various projects with you, but unfortunately I'll have to jump out fairly soon. I have another meeting coming up. But thank you for your. Oh yeah, here's just a flow chart that our at Lund University, our research services person put up. Just a flow chart for what needs to be done if you want to make sure that you're doing things correctly. Anyway, thank you. Maybe there's time for a question or two.
Thank you so much. So I can say that at SciLife Lab, we're just starting with all the Nagoya issues. So now we rely heavily on the universities. Just a comment there. The thing is that some countries, they have their own ABS rules prior to Nagoya. So even if you work with samples from some countries that were obtained before 2014, you still need to check on the Access Benefit Sharing portal which rules were in a specific country before the Nagoya Protocol came in place. Do you have any questions, please? Yes.
Thanks. So I just wanted to check if I understood you correctly on the if you are doing a collaboration and your collaborators are the ones who got the permits and collected samples, but unless there is a clause saying that someone else can also in the collaborative team do, for example, the sequencing, we can't do that, perhaps. Did I understand you?
So, I don't know who is being addressed for the question, but I can say that the person accessing the genetic resources is the one who needs the permits to do the extraction of DNA and sequencing of DNA. So not the collectors. It's good if you have a collaborator in the country of origin, and perhaps they can then help to make sure that you have the right permits so that you in Sweden can then extract the DNA and sequence it.
Okay, but then say that you are doing a collaboration with 3 different countries. The sample comes from one, where you have the country of origin, where you have the collaborators, and then you have someone who sequences it from the other countries, and then you are, for example, doing the data analysis. Then everyone, all the parties need to be in the contract, right?
That's the best thing to do, to make sure that all the parties are on the contract. The one thing that I didn't yet touch on, and it's a very diffuse subject and there's no consensus, is DSI, Digital Sequence Information. DSI is not yet under Nagoya Protocol, but there are moves to make it so. So it's kind of up in the air what happens after you've sequenced it and you have AGCT on your computer as a text file. Is that the genetic resources anymore? It's not clear.
Okay, thanks.
Thank you. May I give some extra comments on that? Since I've been dealing with such a situation at my university, and I like you can do either way, but I always try to look for the most efficient way. And we collected samples all over the world. They were sequenced in Canada, and then the information was sent to Sweden, Finland, and US. So we will have like many parties involved and also many countries involved. What we did was I drafted agreements where we put the project as a kind of use the project as a framework, that we are going to do this, and as long as we stayed within the framework, doesn't matter what person or what country who did the actual task, as long as we stayed within the project and what we said that we were going to do, we were kind of fine, and we got plenty of permits doing that. So you can be creative also when applying for permits, because to be honest, many countries out there, they don't really know how this works too. They are beginners, I'm telling you. That can also be a reason why, for example, in Greek, you don't find the right person, because there is really no right person. So if you are proactive and come up with suggestions like, we are going to do this, we want to be compliant, and we want to follow the Nagoya Protocol, and come up with the first draft, you may get it. Or I would even say it's a pretty big chance to make it. So think practically, because we don't want to have endless of different contracts.
Thank you. In the interest of time, we'll have a Q&A session right after this one, just to talk about the legal issues. Is it okay, Jan? Yeah, thank you. Thank you so much, Niklas. I know that you're very busy, but it was really important to know the researcher's perspective, because infrastructure is good, but what researchers think about that is also very important. Thank you so much for your time.
Thank you.
Thank you.